In section 9.8, we begin a discussion of smooth muscle. We remind ourselves at this point that smooth muscle is not under conscious control, and as we look at the cells, we see some very drastic differences relative to skeletal muscle. And so we notice that the cells are fairly small relative to skeletal muscle cells. They're at least 10 times smaller than skeletal muscle cells, and the cells are spindle-shaped. The cells contain a single nucleus versus multiple nuclei in skeletal muscle cells, and smooth muscle cells, interestingly, retain their ability to divide throughout the course of the life of the cell, unlike skeletal muscle cells, which have a very minimal, if any, ability to divide. We notice that most of the proteins that we see here are going to be similar to the proteins that we observed playing a role in the contraction of skeletal muscle cells. We do notice that troponin is absent. Tropomyosin is present, as are myosin and actin. Although we also notice that we have about a third the concentration of myosin and 2 times the concentration of actin in smooth muscle as compared to skeletal muscle. We also see caldesmin present, and so caldesmin is going to be an enzyme that interacts with calcium and helps us to regulate the contraction of smooth muscle cells. The arrangement of the thick and thin filaments, which do exist, being composed again of myosin and actin, in smooth muscle cells is different than in skeletal muscle cells. What we see is that instead of Z-discs, we have these little dark dots that are shown in the diagram here, if you follow the cursor, and here. They're referred to as dense bodies, and so the dense bodies serve as anchoring points for the thin filaments. If we zoom down to this little, or go down to this little zoomed in circle, we see a dense body and we see the thin filaments projecting from the dense body, and then we can see the thick filaments located between the thin filaments on adjacent dense bodies. And so the contraction of smooth muscle cells is relatively simple. If we look over here to the left-hand side of this diagram, we see a relaxed smooth muscle cell. And then we can imagine the interaction between myosin heads and actin that we observed in skeletal muscle to transform the cell from a relaxed state on the left to a contracted state here on the right. The cell bunches up and even squeezes out, the plasma membrane squeezes out a little bit between those dense bodies as the smooth muscle cell contracts. It's also worth noting this, that the length-tension relationship in smooth muscle cells, although we don't have a graph of it here, is similar to what we observed in skeletal muscle, but the optimal length, or let's say the length at which a skeletal muscle cell can generate tension, is much broader than what we observed in a skeletal muscle cell. So in other words, a skeletal muscle cell, as we can see down here, can be stretched or it can be compressed, and it can more effectively generate tension in that stretched or compressed state relative to a skeletal muscle cell. Now, interestingly, in the case of smooth muscles, the regulation of contraction is a result of actions that occur in the thick filaments with the myosin proteins, as opposed to skeletal muscle cell, in which we saw regulation of contraction was the result of the thin filaments. And the steps that lead to contraction are as follows. The first thing that happens is that calcium levels go up in the smooth muscle cell. That's identical to what we observed, of course, in the case of skeletal muscle. And when calcium goes up, calcium binds to the protein calmodulin that we discussed previously. Once calmodulin is activated, it activates a currently inactive, before calcium rises, myosin light chain kinase. This active myosin light chain kinase, which is bound to calmodulin, then uses ATP to phosphorylate a myosin head. Once phosphorylated, the myosin head on a thick filament begins to undergo power strokes. And so ATP plays 2 roles here. It initiates the movement of power strokes in a myosin head in smooth muscle, thereby initiating contraction, and just like in the case of skeletal muscle, 1 ATP is necessary for each power stroke that the myosin head undergoes. Contraction will continue and the use of ATP will continue until myosin light chain phosphatase comes along and dephosphorylates the myosin head. What we notice in the case of smooth muscle contraction are a couple of differences relative to skeletal muscle. One is that the speed of contraction in this case is much slower than the speed of contraction we observed in skeletal muscle. The form of myosin that we have here operates at a speed that's 10 to 100 times slower than what we observed in the fastest myosin proteins that are found in skeletal muscle, the type 3 fibers. We also notice that we have little or no fatigue here. This contraction is occurring at such a slow rate that fatigue tends to not be an issue in the case of smooth muscle contraction. We also notice that relaxation occurs in the case of smooth muscle only when, as we mentioned before, dephosphorylation occurs as a result of myosin light chain phosphatase. Myosin light chain phosphatase is always active, and it's only in periods when the calcium in the cell increases that calcium-mediated phosphorylation exceeds dephosphorylation. So the control over contraction here is not as rapid as we observed in the case of skeletal muscle, but an influx of calcium leads to an increase in phosphorylation, which leads to contraction, and then the removal of that calcium leads to a decrease in phosphorylation, and we still have that constant dephosphorylation mediated by myosin light chain phosphatase, and so relaxation occurs as those calcium levels drop. The last thing we want to note is this, that something referred to as latch state can occur in the case of smooth muscle. This is a situation in which the myosin heads cycle through the power stroke very, very slowly. And so the dissociation of the myosin head from actin occurs at a very slow rate. This allows the smooth muscle to maintain a great deal of tension with very low expenditure, a very low expenditure of ATP. This is useful in parts of the body, such as in the GI tract, when we think about sphincters. And so those sphincters have to maintain a contracted state over long periods of time. The fact that smooth muscle can undergo a latch state enables those sphincters to maintain a contracted state while spending very little ATP. In the case of smooth muscle, the calcium that leads to contraction comes from two sources: the sarcoplasmic reticulum and extracellular calcium. What we notice is that the sarcoplasmic reticulum in smooth muscle is less developed than in skeletal muscle. Also, we notice that there are no T-tubules that allow action potentials to proceed from the outside of the muscle cell plasma membrane to the inside. So what we see is this, that in the case of sarcoplasmic reticulum that's located near the membrane, calcium is released from the sarcoplasmic reticulum following the arrival of action potentials in a manner similar to what we observed in the case of skeletal muscle and the interaction between the T-tubules and the sarcoplasmic reticulum there in skeletal muscle. But in other cases in smooth muscle, what we see is that some of the sarcoplasmic reticulum is located near the center of the cell. So in this particular case, calcium is released in response to second messengers that are initiated either as a result of action potentials or ligand binding, for instance hormones or paracrine factors, to receptors that are found on the plasma membrane of the smooth muscle cell. So this is certainly different than what we saw in the case of skeletal muscle, at least in terms of the release of calcium as a result of second messengers. It's also worthwhile noting that, even at this point, that this means that contraction in the case of smooth muscle is going to be much slower than it was in the case of skeletal muscle, because of course the action of a second messenger is going to be much slower than the electrical activity that we observe that exclusively controls calcium release in skeletal muscle. In the case of smooth muscle, extracellular calcium also contributes to the calcium that leads to muscle contraction. And so there are both voltage and ligand-gated channels, interestingly, located in the plasma membrane that allow the calcium, which is 10,000 times more concentrated outside the cell than inside, to flow into the cell. So both of these mechanisms, in the case of smooth muscle, calcium from the sarcoplasmic reticulum and calcium from outside the cell, play a role in the elevated concentrations of calcium that lead to smooth muscle contraction. What we notice is that, just like in the case of skeletal muscle, smooth muscle relaxes when calcium levels drop inside the cell. And so this is accomplished, similar to what we saw in skeletal muscle, by the active transport of calcium both into the sarcoplasmic reticulum, that was like skeletal muscle, and this is not like skeletal muscle, but in the case of smooth muscle, we also transport calcium out back to the extracellular fluid. And like we mentioned a moment ago, this means that the twitch speed in smooth muscle is much slower than in skeletal muscle. A twitch lasts several seconds in the case of smooth muscle, whereas it only lasted a fraction of a second in the case of skeletal muscle. We also notice this, that in the case of smooth muscle, graded contractions are possible depending on the concentration of calcium that enters the cell. This is different than what we saw in the case of skeletal muscle. In the case of skeletal muscle, a single action potential typically leads to the release of sufficient calcium to bind to all of the troponin, temporarily moving tropomyosin out of the way. And as we talked about, all the myosin heads may or may not bind to actin in that time. But nonetheless, typically we have saturation of that troponin for at least a few milliseconds. In the case of smooth muscle, it's quite a bit different. And so we don't saturate calmodulin, the calmodulin binding sites, as we first let calcium in the case of a smooth muscle contraction. And so the more calcium we let in, the more calmodulin is activated, and therefore the more myosin heads are binding to actin. This allows us to control the strength of contraction in smooth muscle by regulating the concentration of calcium that's found inside the smooth muscle cell. A very distinct difference that we see between skeletal muscle and smooth muscle is that multiple stimuli can activate smooth muscle. All of these influence calcium concentrations inside the cell, as you know from our previous discussion, but there are 5 different stimuli that can activate smooth muscle contraction, and there was only one, of course, the arrival of an action potential at a motor end plate, that could initiate skeletal muscle contraction. So these 5 stimuli are listed on the screen: spontaneous electrical activity, neurotransmitters, hormones, local changes, and stretch. And we'll talk about them over the next couple of slides. We begin with spontaneous activity that leads to smooth muscle contraction. Interestingly, certain types of smooth muscle generate action potentials with no input at all from the nervous system. These smooth muscles exhibit what's referred to as a pacemaker potential. A pacemaker potential is illustrated in the bottom left-hand side of our slide. And what we notice is this, that in a pacemaker potential, we have a charge inside the cell that slowly depolarizes up until we get to threshold, and then an action potential occurs. And then we slowly depolarize once again in an action potential, slowly depolarize again in an action potential. And so this is referred to as a pacemaker potential, and some smooth muscle cells exhibit this type of membrane charge. This is referred to as a pacemaker potential. Another possibility is something referred to as slow waves. And so in the case of slow waves, which is depicted on the left-hand side of our slide here in this figure, we have charge inside the cell that goes up and down and up and down. And so with no external input, then that's all you've got. So you're never reaching threshold and never conducting any action potentials. But what we see is this: if we superimpose some external stimulus, perhaps the release of a hormone or perhaps stretch on top of that, then what we see is that at certain times, of course, at the top of one of these waves, you'd be much closer to generating an action potential than you would be at the bottom. And so the end result of that is that you have these little bursts of action potentials that occur. If the external stimulus, the excitatory stimulus, is applied here, you get 3 action potentials. It's applied here as well, the external stimulus is, but you don't get any. And so you have these little bursts of action potentials that occur depending on the charge that's present inside the cell at the time the external stimulus is applied. We see both of these types of electrical activity found in smooth muscle cells that are located throughout, for instance, the gastrointestinal tract. The small intestines, for instance, tends to contract spontaneously and constantly, even in the complete absence of any input from the nervous system. The second factor that we see leading to smooth muscle contraction are nerves and hormones. And so the junction between nerves and muscle, nerve and muscle, is very different in smooth muscle as opposed to what we observed in the case of skeletal muscle. In the case of smooth muscle, what we see is that smooth muscle is only innervated by, as you know, autonomic nerve fibers, and those autonomic nerve fibers release neurotransmitter in these little structures referred to as varicosities. And so varicosities are enlargements along a neuron. If we look down at the bottom of our slide and follow this neuron, we see that we have an enlargement here. It's a varicosity, a varicosity, a varicosity, a varicosity. And so an action potential would proceed along this neuron, and each time that it arrived at a varicosity, it would release neurotransmitter, release neurotransmitter, release neurotransmitter. And so you can see that this single neuron reaches a large number of cells in this sheet of smooth muscle. It's also true, although this is not illustrated in our slide here, that one cell may be influenced by multiple neurons. Most commonly, you would expect a single cell to be influenced by both sympathetic neurons and parasympathetic neurons. And so the overall activity, the overall contraction observed in that muscle cell would be the result of the summation of those two signals. We also notice this, that neurotransmitters in smooth muscle may increase or decrease activity. And so that is, of course, different than what we saw in skeletal muscle, where the relationship is fairly simple. The arrival of an electrical signal in skeletal muscle always leads to excitation. We also notice this, that the same neurotransmitter may have different effects depending on where the smooth muscle is located. A good example would be norepinephrine. Norepinephrine, interestingly, enhances vascular contraction but reduces respiratory contraction. It does so by binding to different receptors. Alpha receptors located in blood vessels bind to norepinephrine and cause contraction, whereas the binding of norepinephrine to beta-2 receptors in the airways leads to reduced contraction and relaxation of those airways. This, of course, is adaptive because it allows the constriction of blood vessels around the gastrointestinal tract, reducing blood flow to those areas in response to a fight or flight response. But it allows relaxation of the airways, and so more air makes its way into the lungs in response to that fight or flight response. Hormones are also may have receptors present on the plasma membrane of smooth muscle cells. Those, for instance, smooth muscle cells found in the uterus are responsive to various hormones, most notably oxytocin, which leads to depolarization and contraction. The fourth factor that we want to talk about that leads to smooth muscle contraction are local factors. And so local factors include things like paracrine signals, changes in pH, changes in oxygen levels like we observe in the respiratory system, changes in carbon dioxide levels, also respiratory system, changes in osmolarity like we observe in the renal system. All of these factors lead to local changes in smooth muscle contraction state independent of any hormones or nerves. Nitric oxide is one of the most common paracrine signals that leads to smooth muscle response. In this case, it's typically smooth muscle relaxation. Nitric oxide is released from some neurons as well as some endothelial and epithelial cells. It has a very short lifespan, so it can only act locally, and it causes smooth muscle relaxation. Nitric oxide will be an important vasodilator when we discuss the circulatory system here in just a short period of time. the fifth and last factor that can cause smooth muscle contraction is actually stretching. And so in the case of stretching, we see that we open mechanically gated ion channels that therefore allow the entrance of sodium and or calcium, which changes the charge inside the cell and initiates action potentials inside the cell. The results of this typically oppose stretching, and so we see smooth muscle cells of this nature in, for instance, the stomach and the bladder. And so as stretching occurs, activation of contraction is observed to oppose that stretching. As we close our smooth muscle discussion, we want to mention this. There are two types of smooth muscle cells, and most smooth muscle consists of both of these cell types. So one type of smooth muscle is single unit smooth muscle. And so in the case of single unit smooth muscle, we have cells that are connected via gap junctions. We have pacemaker cells that are distributed throughout the sheet of smooth muscle. And so this type of smooth muscle would exhibit spontaneous contraction independent of the nervous system. And so we see this type of smooth muscle in the intestinal tract, as we mentioned previously. Small diameter blood vessels, the uterus, for example. The contractile activity is modified by nerves, hormones, and local factors. Oftentimes it's stretch sensitive, but the overall initiation of contraction is independent of the nervous system. Secondly, we have multi-unit smooth muscle. And so multi-unit smooth muscle, interestingly, doesn't undergo action potentials at all, but simply the levels of calcium that are found in the cell are altered, and that determines the overall contractile state. But there are no gap junctions here, so each cell responds independently, and the signal is not transmitted from one cell to another. These are typically not stretch sensitive, and the overall contractile state of smooth muscle, multi-unit smooth muscle, is primarily the result of nerves from the autonomic nervous system that are providing input.